So could
Okay.
you please tell us about the format for the exam as well?
Okay. I'm going to give a take-home exam because an online exam would mean the same thing.
Yes, sir. So
But
the question that we had was whether it would be in the same slot that is assigned to SBL 702, or since it's a take-home exam, it will like occur after the minors.
normally, I'll just give it just during this minor period, and I'll have it at the end of the minor period. So, which means 21st, right? Which is the class working day. So maybe 22nd, which is a working day, that will be the last. So 21st is the last time that you can do this and submit it. It'll be a little longish so that you have to work on getting all the details from various internet sources and then compile your answer. So it's
Okay,
going to be
sir.
like that. Okay. So it will be something like an application of whatever we are learning till this point. Right?
Yes, sir.
Okay. So this was the last uh, slide that we discussed in the previous lecture, and we went through the whole analysis of how these isoclines are drawn, and based on the shape of this face plane portrait, we can decide whether or not um, a system is going to be stable or unstable, given the initial conditions that are used to simulate the model, right? So any questions up to this point that we had in the previous class? If not, then I'll move forward and discuss a few other things. So this is, and I'll just give, there are 3 more examples that I'll show, but we'll not go through every one of those. So this is an example of how, the DNA repair mechanism works. So you have an activation, for example, shows you the activation of a tumor suppressor protein P53, which transcribes the genes. And this would induce cell arrest, DNA repair, and apoptosis. And then P53 again activates, this is the direction in which it goes. This activates this protein, MDM2 protein here, and which in turn negatively regulates this. That means somehow it will come back, and but it's not shown here. So you get degradation happening here, but this one with negatively regulate means that it has to go back and negatively regulate the activity of P53. And there's a negative feedback loop formed by P53 and MDM2, which also includes a significant delay because of this entire process that takes place here. So there is a negative feedback loop formed between this and this, and because of that, you have delays that will occur in this system here. There is also a path that shows that how this protein essentially will actually help in ubiquitination and degradation of proteins. So so there is a stability analysis done for this. Now, question number 1, which I given, is of course the van der Waals oscillator, and the question number 2 is based on this. So what this model does is looks at these 2 variables, which is the first one is P53, the second X2 variable is MDM2, and these are the parameters. The descriptions are given here, and then based on that, you can simulate this model. And once you simulate this model, you can find out the characteristics of whether or not this is stable or unstable. Now, I've left it at that because this was supposed to be a problem to be done by the students. So you could try that out by yourself, and I will not go into the discussion of that. What I'm going to discuss is the trip operon. And the reason I'm doing this is because it's slightly different in terms of the way we look at And we'll come back to this later on also when we're doing transcription control. But right now, I just wanted to give this as an example of modeling. So how many of you are aware of what the trip operon operates? I mean, how does it work? Yes, how many of you know? Nobody's answering.
So we briefly told about it when we were taught about lac-operon, but not in.
Okay.
Yeah, the same, sir. I have a brief idea like how it like kind of operates.
Give me a second. Sorry, I don't have a glass of bottle to pad on the screen. So anyway, let me discuss this. Okay. Now, the lac operon is a negative control operon, and the beta galactosidase, for example, is a positive control operon. The trip operon is essentially an attenuation operon. That means it will have a graded response to the amount of tryptophan that is present in the system. And the example that is picked up is for E. coli. And this trip operon has, these proteins that have to be synthesized. This, of course, is a leader sequence, but we'll not talk about that. We'll talk about that separately while we're discussing the control. So this is where all the regulatory control takes place, these two together. But these are the proteins that are to be synthesized. So E, D, C, B, and A. So the reverse way. And so it shows you that these two components together gives you one here. This one gives you a separate protein. And this one then gives you another one. And you require all of these to essentially get tryptophan. So this shows you the enzymes that would be acting on these metabolic pathways and how these metabolic pathways would then act one after the other to give you this one. Okay. Now, modeling all of this would be quite difficult. There are models which do account for all of these, but the model I will be discussing will be only considering this whole group to be one consolidated enzyme, which will take chorismate to, let's say, tryptophan. That kind of a assumption will be made. Now, what it has here is an operator region, and also what it has here is a leader region here. And the leader region has a embedded attenuator. I'll make the discussion a little quick because we are not really going to be discussing those things which go into all the details of the tryptophan operator. But let me know if there's some kind of importance or some kind of confusion is there. Okay. Now, what happens is that this happens for quite a few of the other amino acids also. Histidine is also another one, and there is one or two other which are also working with this mechanism of attenuation. So what happens if you add tryptophan to a growing cell for cultures? It essentially stops producing this enzyme, which leads to the synthesis of this pathway. And attenuation is not regulated by the end product, but the operon is not regulated by the end product, but by another mechanism which is part of the mRNA structural changes that occur. Okay, I'll come back to this to this first slide here. The reason being is that there are two things to keep in mind. I don't have all because this is supposed to be a shorter version of the entire length of slide that I discussed. But anyway, look at this operon site here. So when you have excess of tryptophan in the system, then there's a repressor molecule which is there, and this repressor molecule binds to the tryptophan, and together when it comes here, it will essentially inhibit the site from which the ribosome, the RNA polymerase can bind and then synthesize the mRNA. So that's where the main controllers. So if you have a lot of tryptophan, then there's no need to produce tryptophan, because as you can see, to produce tryptophan, you require five enzymes, 1, 2, 3, 4, 5, of which 2 will form together as dimers, and then 1 is independent of that. But this one acts on 2 pathways. Now, so if this operator site is free, then the RNA polymerase will bind here, and then it will go forward to synthesize the leader sequence as well. And that's where this gets interesting, because the leader sequence, if you look at the entire synthesized component of it, then it has 2 configurations that it can acquire. And that is because it has regions within it, which are called regions 1, 2, 3, and 4, which are complementary, which have complementarity. The complementarity is this way, that 1 and 2 are complement to each other, 2 and 3 are complement to each other, have complement regions rather, which can allow it to bind. And then 3 and 4 also have complement regions. So you can have 1, 2, 3, 4, because 1 and 2 have complement regions, and 3 and 4 also have complement regions. So you have this configuration possible, 1, 2, 3, 4 configuration. And the other one is 1 and 4 are vacant, but 2 and 3 will bind and give you this configuration. Okay. So when you have this structure, you have what is called a terminator sequence. And when you have this structure, we have an anti-terminator sequence. Okay. All right. Now, let us look at how this works. So very simply, it works like this, that if there is a certain threshold, of course, if the tryptophan that is present is below the certain threshold of that required by the cell, then what happens is there will be the initiation of synthesis for this, because what will happen is the tryptophan will no longer bind to the repressor, so the repressor site is released, and when the ribosome, when the RNA polymerase will come, it starts synthesizing it. Now the RNA polymerase is leading the ribosome on the this RNA polymerase, so this mRNA that gets synthesized is getting synthesized from the RNA polymerase that is on the gene, and the ribosome is on the mRNA, and that is going to be synthesizing the protein. Now, so you have a strand of the DNA on which you have the ribosome, and the mRNA is synthesized, and this one is following. So the ribosome will be following the RNA polymerase as it moves forward. Now, if you have high levels of tryptophan, then essentially there is a region in this leader region where there are 2 successive codons for tryptophan to be incorporated. Now, if there is not sufficient tryptophan, and if there is sufficient tryptophan, then this will essentially translate very quickly and move on to block of this part of the mRNA. So while 3 and 4 is getting synthesized, this one, the 3 and 4 will form together this loop, and this loop will not. This loop will be forming in such a way that it essentially form this terminator site. Now, when you have the other situation where you have low amounts of tryptophan, then it was on the region 1 where you require tryptophan to be brought in. It halts there, and so because it's halting here, 2 and 3 when they form, 2 and 3 because 2 is vacant, ribosome is not there, so this will form a loop. I'm sorry, this will form the complementary structure, will bind and form the structure here, and then this structure that is there will actually allow this ribosome to continue and form the full transcription, whereas this will not. Okay. Now, so much said, you need to go back and perhaps look at this one more time. But the reason I am presenting this here is because this thing can be modeled, and the way you model that is by looking at the basis of what is happening here. So this is the wiring diagram that I was telling you about. So what happens is you look at the mRNA, And as the mRNA is getting synthesized, this mRNA, the dot means it can also get degraded later on. Okay. So the mRNA, if it is getting synthesized, it will activate the enzyme, and there's the pool enzyme I was telling you about. And that will also activate the synthesis of the RNA polymerase. So this is how it's being shown here. Sorry, the tryptophan here, it gets synthesized here. Now this tryptophan that is synthesized here is going to be having a negative feedback here, as well as it will go and bind to the receptor, and the receptor will go back to the operating site and then inhibit that synthesis. So that's the repression that takes place. And when it is on the forward direction of this one, it essentially will regenerate the receptor and then produce the tryptophan. And the tryptophan that is being, it goes out, and the tryptophan that has been synthesized here will get consumed. Okay. So the synthesis, that's the way this scheme is shown. This is the activation, this is the inhibition. Now, the way you write out the model is something like this. So if you were to look at, so the rate of synthesis of, let's say, P is dependent upon the rate of synthesis of this is the rate of synthesis of the mRNA, right? So X is the mRNA. So the rate of synthesis of the mRNA, which is here, depends upon its intrinsic rate by which it will synthesize, and that is given here. Okay, and there's a rate at which it's going to get removed or get inhibited, and that is going to be, sorry, where am I here? So that's going to be this one inhibition that comes here, and there's own degradation that takes place there. So two components are there. So likewise, you can go back and you can actually apply this, and when you do this, you can actually work out the model and find out what are the dynamics. Okay. All right. So that is the procedure. So any questions on this so far? Yes.
No, sir.
Huh? So everybody understood everything, is it? Because I was thinking I was not doing a very good job in explaining. Anyway, the reason is because I'm kind of cutting it short, because there's much more to it than meets the eye here. But I would just wanted to illustrate the procedure that is there, that you're taking something which is completely biological in understanding to something that is completely mathematical in your understanding, right? And there are steps to it. So the first thing is that you break it down into steps that are understandable to you in terms of mechanism, and then you use that and then try to develop something as we call like an information flow or a wiring diagram. And then based on that, you can now put in the parameters, and looking at the way the reactions usually work, you can essentially write out these components. Okay. All right. I'll leave it at this because this is actually a solved problem in one of the books that I have been referring to. So, but anyway, the solution for this, you would anyway have to essentially code it. Otherwise, you can't do it. Okay, I'm giving you another example, and I'm going to skip this one here, and I'm going to talk about chemical oscillator. Okay, and this is something that I'll also illustrate on the MATLAB because it has a very rich dynamics. So that's the reason I'm choosing this. So I talked about chemical oscillators before, and I talked about limit cycles also. And these are some things which are very typical of nonlinear systems. You will not see limit cycles in linear systems. Okay, and these exist very well in biological systems, and some are robust to perturbations, others are not. And the limit cycle will actually vary. You can get certain situations where you have a limit cycle, and in other situations you will not get the limit cycle. So depending on the initial condition, you will be either, because if you're within the limit cycle and the limit cycle is actually stable, it will go into a stable equilibrium, then that works out. So, I don't see that. There was another set of equations. Anyway, let's use this set of equations here. So the phase field analysis of this is that you have three equations. So you have X, which is reversal going to A, B, which is permanently going to irreversible going to Y, and then X and Y gives you something else. And then that X again has a reaction which is based on, which gives you A. So that's a loop that forms there. And by the law of mass action, you can easily see that if I wanted to write the rate of change of X, then the rate of change of X is dependent upon this equation first of all. So there will be K3 times the left-hand side, which is, there are two molecules here. If you take the mass action kinetics here, then that will give you X squared Y. And then you have this one reversible here, one K1I. And so this is going in the reverse direction. So it's producing X, so that will be plus. So this gives you a plus sign here. And then it is getting removed by this K1 reaction, so minus K1. And similarly, you can look at Y. So that will give you, it is being synthesized by this reaction, and it is getting removed by this reaction. So you get those. And some parameters which are used in dimensionless form, you can, for this kind of work, just to illustrate, is taken as, set all this to 1, set this to 0.1 and 0.2 and see what happens. And so I'm going to show you how to use this in MATLAB. And I kind of, this is not the one, I've done this. Again, this one. Okay, I'll just use this one here. Can you see this on the screen, the MATLAB also?
We can see it, sir.
Yeah.
Yes, sir.
So so there is this one, this unit is essentially a computational unit that is there in the library here, library browser if you go. And if I go into, I think, continuous blocks, if I'm not mistaken. User driven function. So what I've used is this one is system. This one, no, not this one. I think this. Let me just see if this is the one that comes here. Oh, this one. There's another one here. So I've used this one. So when you open this, you get this function here. Okay, and I'll show you how I use that function here. Oops. Where did that go? Okay, maybe this is the one where I wanted to show you, so I'll go back. So, yeah. So for this one, I actually embedded this inside one of these systems. I used the system and then embedded it here so that I can add more features on the outside. And then you go into this, and the function that I had, I defined that as x1 dot, and then the function on the right hand side are the two variables that I'm using. So I define the initial conditions and the parameters, and I look at So you can see that this is that x3, k3 into x1 to the power of 2. So x to the power of 2 and y. Then k1 I star a, that was the one that you had, minus k1 x1. And then x2 is k2b, which is this constant here, which is dependent upon what I showed you earlier. And then this is the same one, negative sign. And so that's the set of equations. Very simple in this case here. Okay. So then I just wanted to see how this would work out. So I'm going to just delete these two from here. And if I just run this, so then I can see this graph here for, so this one is 1. So I have to go back and see what 1 is. 1 is x1. Okay. And then 2 is x2. This is how it looks just by itself with respect to time. And then 3 is the plotting of x1 versus x2. So, and you see that that is going into some kind of a stable zone. So this is not really an oscillation, but it is essentially going to a stable node. But if you continue exploring the parameter space, you'll get one location where you'll start getting an oscillator, which is a closed curve, which you'll get, which is called a limit cycle, and you get a closed curve on this plane. So you can see that this can be very easily worked out. And I'm just going to come back here to my presentation. And so ideally, this is what you should be getting, y versus x. So I got something which is inside this. Okay. So something that would have been inside this, and is in this case is going to the limit cycle. So it will move outwards. So it means that it's a node which is unstable, but a limit cycle which is stable. Because now the profiles from the outside are going into the limit cycle, and the profiles from inside are also going to the limit cycle, which means that the limit cycle is a stable structure or feature, and the node which is inside is going to be unstable. And that you can work out by the direction which is there from the initial condition, whether it's going in or out. All right. Any questions on this? There is a MATLAB part of it, so you need to go back and work this out. So would you be able to do this by yourself? Well, how many of you are really looking at this now?
So you can try on your own first.
Yeah, but you see, that's what we've been saying for the past few lectures. So people do have to work this out. Okay. So there are more. I've got lots of these examples. You can look at bacterial chemostasis. That's one model, which could itself be quite a lot of work to do by something. Then there's another one which is on the MAPK cascade, and so there is this basic structure that comes up from the signal transduction in xenobus oocytes, and how that starts, giving you a cycle like this. And so you can see here that now it's like far more complex because you have phosphorylation taking place on the NEPKs, and then the P42, which is part of this whole cascade in the case of the oocytes, that also has to be double phosphorylated. When it's double phosphorylated, it activates this, and that together will give you some kind of feature that is required here for the synthesis of the oocytes or the degradation of the oocytes, something, whatever is the problem there. Okay, and you can see that now the problem becomes quite difficult because now you have got 10 reactions, 10 parameters, sorry, and 1, 2, 3, 4, 5, 6 reactions, and one balance equation, one constraint. And so as you start building up, this will go more and more. It gets quite complex. And there are various places where this is applied, and to understand various kinds of systems. Even in bioreactors, you can get behaviors like this. So how do you define a general system? You essentially draw the boundary, look at the inputs and outputs, and then you define the equation. So I'm not going into this because this is not part of what we do, but those who are in biochemical engineering or are from the biochemical engineering department would be able to appreciate this, that you can do both energy and you can do energy balances, you can do material balances, as well as you can do balances for the cell by itself. The cells that are there, you can do completely a different kind of analysis for the what is happening to the cells inside this, besides the material balances that take place. Now, also, the behavior is very different for different situations, and you can get oscillatory behaviors. In large-scale behaviors, you can get predator-prey models where there will be oscillations. You can have other kinds of behaviors where you have a point from which the system evolves. It could be an unstable behavior like this, where it essentially goes out to two other points. So you have one node which is unstable, but you have two other nodes which are stable. So this will be a bistable structure. So even if you're here, you'll either be in this solution or in this solution. And then you can have other kinds of behaviors where you have far more nonlinear behavior, where one behavior will tell you tumor growth, and the other one will give you the other growth which takes place here. There are other situations here which we talked about earlier, and the general format in which you represent these equations are given here. So if you have linear system, then you have a matrix which is a constant matrix. If it is time dependent, well, it will be AT, but it is separable from the variables, whereas in this case you can't. And so you will have a kinetic structure Fx and a transport structure Gx. Okay, all right. So that said, and so there are other models. You have so many different kinds of models. I'm not going into details of this now for this lecture. Now there are other things that we can do, and I wanted to illustrate to you how if you had a problem at hand, and how would you go about essentially solving it. So let's say I give you a problem, just like I give you that keg pathway. I give you another pathway, and let's say this is an exam, right? So in an exam, I could give you very easily a keg pathway, and I'll ask you to just work out the entire model and tell me what the dynamic stability is for the system, right? It could be very easily done. So, but I'm just going to illustrate that is when you're doing a proper research problem, then there are other things that need to be accounted for. And this one I'm going to discuss in terms of methionine synthesis. So when methionine was being synthesized, there was a series of these CSTR experiments that were done to obtain the kinetics of the process. Okay, so by doing the CSTR experiments, you can extract out the parameters, and broadly based on whatever is normally used in reactor kinetics, you could define a model. And if you did the statistics correctly, then you would be able to actually plot variations of oxygen and dilution rates and the influence of that on the synthesis of cell and the synthesis of methionine. And what you'll find is the scale that is shown here, the biomass productivity and methionine in gram per, this should be gram per, what is this? Gram per liter productivity, gram per liter per hour. So that's the productivity shown here, but this scale is showing you that the biomass is going to be highest at this value here. I got it incorrectly done here. I don't know, it's okay. So the units should be gram per liter per hour. Okay, fine. So you can see that this would be this cell mass, and so I'm not very sure whether I got the units right here. Huh? So I'll have to check and come back and tell you. So I'm not very sure of the units, but anyway, look at the profile. So it essentially shows you that you're going through a maximum at this point. Wherever the maximum of the biomass is, you also have the maximum for the synthesis. So it means it's a growth associated production. And we can ignore this figure for the time being. But what you see here is that you can actually have the plot from various reactor measurements, and you can see two different cases with cysteine supplementation and without cysteine supplementation, and you can see that the productivity changes. In one case, you'll have high productivity. In the other case, you'll have a slightly low productivity. Anyway, so using this, you could do the flux analysis like I told you earlier, and you get a flux map. And then you find that, well, you get this very high concentration predicted for methionine. Well, however, in this reactor, you will not get more than 1 to 2 grams per liter. So why is it that you are predicting a very high quantity here, but you're not getting that same quantity when you're synthesizing this by a reactor? So that is the problem. So the research problem is, so what is the reason why this is happening? Can we find out some way of explaining this difference that is occurring between the prediction that I'm making from the flux map and my actual experiment that I see here, where I'm getting about 2.3 grams per liter as a maximum amount of methionine? Okay, so that is the question that I'm trying to answer. So what you do is you know, I mean, we know the limitation is because that what happens here is that you're looking at steady state assumptions, whereas if you look at systems approach, you don't do that. So you're going to reconstruct all of these using the control structure that are present. So look at the control structure that is here. So what you see is that for this organism, C. glutamicum, there is a repression that is here, and there are inhibitions that are here. Okay. So now, based on this study that was done, it was realized that there are 7 ATPs that are required to synthesize methionine from aspartate. Okay, and therefore, so if you forced a mutation, which is what we have been doing in the case that I showed you, because we mutated the organism to get high quantities of methionine. So what is really happening? So that is the analysis. So I'm asking the question in a different way. Same question is being asked, and so what you do is, to do that, you essentially you do a model, and in this case, to illustrate the point, we are using the same Simulink that I showed you earlier, and inside the Simulink, so you'll have this inside the system that is embedded. You'll have this embedded function inside, and let me tell you that this enzyme block has other components inside it. So, for example, you're looking at delays that are occurring across transport across the membrane. You're looking at delays that are occurring, because of the synthesis that is taking place here, and you're also looking at delays that would occur because of actual synthesis that's taking place. Now, what we normally do is we say that the enzyme concentration is fixed. But here what happens is, because we're able to include the mRNA synthesis rates, we say that the enzyme concentration is not fixed. So that is going to have a variable. That is the mRNA synthesis here, and that is the transport that is occurring across. So the enzyme synthesis that is there is taking place based upon what the response is to other cues that are coming into that block. So that was a block, and this is the enzyme block, and inside the enzyme block you get all of these cues, and so therefore this block is telling you that the rate of synthesis of the enzyme is dependent upon other factors, and therefore the rate of production of some compound from some substrate. So it could be aspartate semialdehyde, for example. You would have situations where this variation would also depend upon what is happening inside this. Now, if you converted that diagram which I showed you earlier here, if I were to use, instead of writing these numbers, so if you just looked at this sequence of reactions that are there, and if I were to just remove this letter and just put a circle here, then that would be the equivalent Bayesian diagram. So it would be something that leads into a circle, and then another node leads to another node, and go downwards as we go. So So what I'm doing here is that, I'm asking this question. So actually, when I come to this block, to fill any of these blocks, which is this block here, I have to really know all of these parameters. And all of these parameters have to be very specific to the organism. So what I have to do is I have to go to KEGG and look out for this pathway. Then I have to go to each of these enzymes that are there, and I have to obtain all of the details that are there for these enzymes that are present. And to pick that up, and then normally what you have to do is you have to build an Excel sheet where you essentially code all of this, into it, so that you know where the information is and how you can use that information. So, so if you want to use the Excel sheet and then automatically populate this one here, you can do that also. But then in that case, you'll have to have a manner in which you will have to show the reactions, and based on the reactions, you put the parameters that are required for that reaction. All of that is possible. Now, what you do is you essentially build this up, and you'll get something like this, which is quite complex. Now, I'm not going into the details here, but you could put input. So let me do one thing here. Let me see if I can expand this here further. And let me show you here. So you can see the spark can be something that goes down or something that goes up, or you can choose some other kind of input here, or it is constant. And you can see that it has inputs coming from other components. So these are inputs coming from lysine block, input coming from the ASD block, and so on and so forth. And you can see that it's quite complex. You can see how many inputs are there here, because that's at the node. So you can see that it has so many different inputs that are coming in, right? So it's quite complex, isn't it? So when you build this up and you look at the concentrations of time profile concentrations, you'll get concentrations of this kind, which is essentially looking at some simulation time here, maybe over some minutes. So here you're looking at 2 hours or 3 hours of time here, and then you could look at all of those intermediates that are appearing in the pathway. And then if I ask the question, what happens because we have forced a mutation on it, then you see that if I only had a lysine mutant, that means it doesn't produce lysine but requires a lysine as an auxotroph, or I could have a threonine as an auxotroph, or I could have both of them. So this is a wild type organism, and I can see that its response to the production of methionine is quite stable. But if I look at the methionine tRNAs, actually really the concentration of the anyway, we'll look at that situation later on here. So we look at the concentrations here, and you look at the concentration of homocysteine on this side. If you look at both of these, you can see that as you increase the number of auxotrophy, so here you're looking at 2 of them, you can see that the behavior is quite changed. So this essentially tells you that you have a difficulty in essentially, you would face a difficulty when you do these mutations, and that could be an explanation why you are never able to reach the highest level of prediction that comes from the flux analysis. So let me sum up. Okay, so I'll go to this slide to sum up. I'm going backwards and redoing this whole thing. Okay. No, it's not this one. Let me do this one. So actually, the main thing that we have to now go through is this. So what I've been talking about is essentially modeling of biological systems and modeling at different levels of biological systems. Clear? That's the first point. And what I discussed in these 3 or 4 lectures is that if you are doing the modeling, you have to have the window over which you will be defining the boundary, and the window in terms of time constants, or the boundary in terms of where you want to enclose the number of reactions that are relevant to the problem. Okay, so you can do 2 ways. You can actually draw the boundary based on the pathway that you want to look at. In this case, I picked up aspartate to methionine, so I picked up that pathway. You could also do it in terms of time constants, and maybe you can look at a broader time constant spectrum. That means you would look at reactor kinetics going all the way to, let's say, enzyme kinetics. So that's another way to look at it. That's the second point. So you could now write ordinary differential equations to describe this. Clear? There is the other method that we talked about, which is that if you wanted to do the analysis and ask the question, where should I mutate an enzyme in a given metabolic pathway so that I get the desired outcome? The desired outcome could be higher synthesis of a certain compound, let's say amino acid or an antibiotic. Then where exactly should I make this mutation? So to answer that question, if you take a pseudo steady state assumption and you do the flux analysis, you get a reasonable answer. So it's a good point to start. But then if you wanted to look at the dynamics of really what's happening and ask deeper questions, such as the one I was asking, that if you are not able to match the result that you are getting in an experiment with that you're getting in a flux analysis, then obviously you will have to look at the regulation that is there. And so to study the regulation, you'll have to go and then do a block in which you would have the enzyme being, the dynamics of the enzyme regulation itself being defined in the kinetics that you're taking into account. So if you do that, then you start seeing deeper questions that can be answered. And one of the questions that we asked is, if you have a mutation in one of the enzymes or something happens to the organism, then what really happens? So we find that if the stability was in question, then obviously it is much less stable than compared to the wild type. Therefore, you obviously will not see a situation where you're getting exactly the same result as the flux analysis result. So this in total is about what we were discussing in this lecture series. And so if you have any questions, please let me know now, and we'll discuss that as a closing of these lectures. Yes. Any questions? No?
Not at the moment, sir.
No questions at all?
Not right now.
So,
yeah.
If possible, could you please give one demo on these kind of models in MATLAB?
Demo. This one, yeah, I have it already. I mean, I have this model built in MATLAB already.
Okay.
So I do not know if it is visible from my MATLAB now. Let me see. I've got a whole bunch of these here. I don't know if that one is here. That one is not here. Okay, I can show it to you. What I'll do is I'll do another. I know that there's a lot of material for one class, or even for these 4 lectures. A lot of material. What I will do is I will spend one more lecture, and maybe bring one example that you can do, and one example that I will show you how to do.
Okay.
So that might be a better thing, because
yes, it will get us some
because
better.
Normally, I don't know if students of this course may not be aware, but those who are in DVB would know that there is a separate course on flux analysis itself. Metabolic engineering and flux analysis. There's one course that is done. So all that we did and compressed in these 4 lectures is really a full-fledged course. And similarly, if I were to take up modeling, I could do a full-fledged course on that. So, so I'm trying to compress a lot of ideas, and it does. But if we can actually do 1 or 2 of these examples by ourselves, we need not know all the details of everything that is part of metabolic flux analysis or part of modeling. But if you can take home just this much, that if I'm given a small pathway, I know how to draw the wiring diagram, and I know how to write the code for this, at least in Simulink. if not in some other coding language, other programming language, then that is good enough for me. That I think is good enough, and that is all I'm trying to achieve in this. So that even so, and I wish we had a little bit more time to spend on this. But let me see if we can do it a little bit later also. But I'll try to do this much, that I show you one example and I ask you to do one example. Okay.
Yes, sir. That will be good. That will be helpful, sir.
Okay, then shall I conclude then today's lecture?
Okay. Thank you, sir.
Meanwhile, you wanted to ask something?
No, sir. I was saying like if you that the same thing, like if you can do that one simple example with simultaneous, then
yeah, I'll do one simple example and then I'll do that complex example, because the bigger examples are the ones that you need to know. And I'll try to bring out the whole folder where I have the data on this, and then maybe show you how it needs to be done. Because there's a lot of data keeping, and also how you record the data, how you keep the data. All that is also important. Okay.
Okay.
So I'll conclude the lecture here, and as always, if you have any questions, you can send an email. We can open discussion session on the chat or on the Moodle, or even in Partus. So that would be helpful. Okay. Okay then. Bye.
Thank you, sir.
Oh,
Thank you, sir.
Thank you, sir.
bye.